Hello, science enthusiasts. Welcome to Spaces Unleashed. Every week on Twitter, we bring an expert to chat live through the Spaces program. And this is bonus content that goes with the Science Podcast. We hope you enjoy the show. All right, it is 7 o'clock. It's time to start. So if you've never been to one of our Spaces Unleashed spaces before, I'll remind everybody of the ground rules and how kind of the Spaces Unleashed works. So I have an amazing guest today, Dr. Samantha Wilson, who's a postdoc and deals with genetics and epigenetics, and we'll get into what that is. And I usually ask enough questions to fit, to take about 15, 20 minutes. And the idea is then we have basically a groundwork for who the guest is and what they are. And then I open the floor to questions from the audience. And I do double check to see who the person is that is asking the question. So if you want to speak and ask a question, I double check to make sure that you're following me, following Bunsen and Beaker. And if you're a really, really new account or your account I don't recognize, I double check to make sure that, you're not a troll account. So sometimes I don't allow everybody to be a speaker. If you're really shy when it's time for questions, you can direct message me the question and I'll read it to Dr. Wilson. So I'll repeat the ground rules every 20 minutes or so. And Spaces Unleashed takes about an hour. Sometimes it's a little less. But the guest is only Dr. Wilson. Do you have about an hour? Is that okay?
Yeah, that's fine.
Okay. Yeah. So after an hour or whenever, or if we kind of run out of questions that people want to ask, then I open the floor if people wanted to ask questions about Bunsen and Beaker, because some people are really curious about what's happening with the dogs that week. All right. So welcome, Dr. Wilson. How are you doing today?
I'm good. How are you?
I'm good. And we were just talking, you are in Toronto, Eastern Canada.
That's correct.
And one of our first questions, just so people have an idea about who you are, could you talk a little bit about what your background in science is?
Yeah, so I completed my undergraduate degree at the University of Western Ontario, and I did that in genetics. And then I subsequently went on to do my PhD at the University of British Columbia, and that was in the lab of Dr. Wendy Robinson. And so I worked a lot with placenta and looking at DNA signatures in the placenta and how that changes with pregnancy complications. And when I was finishing up there, I really became interested in the idea that, there are changes in DNA associated with pregnancy complications, but we can't really sample the placenta during pregnancy because that's invasive. So what I did is I came to Prit after my PhD, and I've started looking at DNA that is shed from the placenta into the mother's blood. And that's what I've been working on the last couple of years.
So were you a kid that was interested in science? Because I have so much respect for people that pursue anything past an undergrad, right?
Yeah.
Were you a kid that loved science? Was something sparked you into go into genetics when you were young?
Yeah, I've always kind of thought about that. And I think I liked playing around and making experiments in the kitchen and trying to getting things to explode. But I wouldn't say that I was a kid that was very obsessed with science from a young age. It probably wasn't until high school when I had really dedicated high school teachers that just loved what they did and got me interested in it. And as soon as genetics was kind of placed in front of me, I was like, oh, I think this is what I want to do. So yeah, I wasn't an early bloomer, I wouldn't say.
I believe we talked before. Was it like the first Punnett square, that kind of genetics?
Yeah, I did. One of my friends likes to joke around that when Punnett squares were given to me, I didn't understand them. I got really frustrated, and she had to explain to me how to do them. And now she's like, "Look at you, 15 years later, and finished your PhD, but I get to claim that I taught you Punnett squares."
And for people that maybe don't know what a Punnett square is, it's like the probability of heritable traits, right, from a mother and father, or
Yes.
male, female, or not even that really sometimes, but it's a heritable traits thing.
Yeah.
Yeah. So you worked with placenta. What, like, were you was that just part of your doctorate to think about fetal development? Were you interested too, or did that, as some people are in their the masters and post or doctorate, they kind of get funneled down a path?
Yeah, well, I wouldn't say I was interested in it in undergrad. I think I spent a lot of time looking at cancer and schizophrenia was talked a lot about in undergrad classes. And then came time to do graduate school. I was like, I want to do something different. And I, came across development and I was like, that would be cool. And then, yeah, I came across Wendy Robinson's lab and I was like, oh, placentas, that I never even thought about that organ. I thought it would be cool at the time. I was like, I'm just going to do a master's. And then I just kind of fell in love with it and stayed for the PhD. And now I don't ever want to leave placental biology. It's a
Can you,
really
can you tell
understudied
everybody?
organ.
Can you tell everybody some things that you found, like for people that maybe have zero base points, like no framework for what you would be studying there?
Yeah, so I look at something called epigenetics, and these are marks that are on the DNA. And some of them are very stable, but some of them change, and they can change with age. They change between the sex of the placenta. So the placenta has a sex that's either male or female, based, and we sex it based on chromosomes. And there's also changes due to environment or whether or not there's a complication. And so I think of these epigenetic, sorry, my dog is wanting attention. I think of these epigenetic marks as kind of, parts of the puzzle where I think when I did I talked about how you can think of it as a musical piece where, all the different instruments are different cell types and they all have different marks on their DNA. And you you can the musical piece will say if it's loud or if it's quiet, and that you can think of each works within a musical piece as the epigenetics. And so we can look at these different marks and uh, whether or not certain signatures are associated with pregnancy complications, and eventually what I was looking at.
So the environment can shape what happens actually with the DNA.
Yeah, the environment can change what's happening with the DNA. There's a whole field in terms of what goes on in utero can change with what's going on in the DNA. But I do want to say that we can report changes, but we don't actually necessarily know what they mean. Changes could be good, they could also be bad. But right
Hmm.
now, we just know that they change.
Hmm. So this is a relatively new field, or is that correct? Like before, it was like your DNA is your DNA and nothing changes it throughout your life unless it mutates.
Yeah, I would say it's definitely come about in the last couple decades. I think it's definitely become more prominent in the last 2 decades for sure. And it's something that we're starting to understand the relationship between all of these marks on DNA and what that means for developing cells, what that means for how we or all our cells interact with the environment, and how our lifestyle plays a role in our DNA and what that means for future health.
Hmm. So is there? I apologize. This is maybe too big of a question. Um, is there a goal for you studying this? Like, do you have something that you're trying to learn or uncover or help with?
Yeah, so I think something that people will hear a lot in I guess in public is the idea of either artificial intelligence or machine learning. So I'm trying to use machine learning approaches on all of these data, looking at these signatures and trying to predict pregnancy complications before they happen. So the goal there is to try to identify pregnancies that are at risk of having some sort of complication, and that hopefully we could maybe monitor it better, or maybe this will lead us to finding new ways to prevent these complications from happening, with the ultimate goal of optimizing the health of the pregnant individual as well as the baby.
So you use the DNA from the placenta as part of this.
Yes, yeah. So actually, the placenta in normal pregnancy is shed into the blood circulation. So the idea is we could take a simple blood test when you're getting blood tests already
Hmm.
from routine pregnancy care, and then we could just look at it and say, okay, there's no mark saying that this pregnancy is going to have a complication, or yes, there is some marks that say maybe this pregnancy is at risk of having some sort of complication, and then maybe we flag that and start increasing the number of appointments that individual has, or increase the number of ultrasounds the individual has, just everything's progressing as we would expect.
Yeah, because I remember like my wife had to go for I don't know how many blood tests when she was pregnant, but I mean, if you're getting your blood tested anyways,
Exactly. Yeah, that's kind of the idea. Is like, well, we're doing it anyways, and if it's just an extra tube, and we could get this information, there's the cost versus benefit there is a no-brainer.
wow, that's amazing. And then the idea is based on whatever you're getting the blood, it can predict or not predict, but it can help prescribe care or even the environment the person is in. That's a marker or that's something that could affect the outcome of the pregnancy.
Yeah, there is an idea that, one of the complications that I study is preeclampsia, which is a hypertensive disorder in pregnancy. And
Hmm.
many of these individuals that develop preeclampsia during pregnancy are actually at an increased risk of developing heart disease later in life. And so that is a question to say, if we have a signature that we could look at and say, okay, you're at risk of developing what preeclampsia, does that also predict whether or not the individual will develop heart disease later in life? We haven't looked at that, but that is a question to be asked, and whether or not these signatures could predict, how well the baby does in terms of if the baby's being delivered very preterm, like how well it's going to do if we deliver early, that kind of thing.
And you mentioned machine learning and
Mhm.
AI.
Yes,
And on the podcast, we talked about how you had to learn to code.
I did.
Could you talk a little bit about? I mean, it's a very difficult concept to kind of like bring it down to a level that I can understand because I know nothing about this. But would you be able to speak to that? Like, what do you mean by machine learning? What do you mean by AI?
Essentially, we have a lot of data. So we generate a lot of data, and even when we're looking for these different marks on DNA, we're looking at millions of marks across the entire genome. And so the idea here is to generate a ton of and look at all of these marks and their changes in association with pregnancy complications, and then to use those marks to predict whether or not a pregnancy is going to develop preeclampsia or deliver preterm. And so, in order to do that, we need a lot of data. And so, the idea of machine learning is basically teaching or developing these models to look at all this data and I guess, identify what marks are the most predictive of a certain complication or whatever you happen to be looking at in this case. So the idea is basically to teach the computers to look at all of our data, pick if signatures or changes in DNA, and then use those changes to predict complication in samples that are not part of the original data that you looked at.
And to give people context, like the amount of data we're talking about is like there's absolutely no way that you one person could do this.
Yeah, no, we're looking at, in some samples there's approximately 27 million sites where we measure DNA methylation, which is an epigenetic, and that's for one individual. So you can imagine having, 27 million values for every single individual, and then we need thousands of individuals, and how much computational power that kind of process would take.
And is there just one more question on that? These programs don't exist. You have to make them because they're every program specific to what you're trying to find.
Yeah, there are programs that we can use that help us with some of these models, but you have to develop each model on your own data,
Oh my God.
essentially. And then once you have that model, you can use it on other data, and that's the idea is to do that. But yeah, you have to do that for your data set first before
Hmm.
you can use it.
Now, one more question before we move on. You mentioned the methylation sites.
Yes.
Um, my background of chemistry, methyl is like a carbon group. Is that true? Am I?
You are. That's exactly what it is that carbon group bound to three hydrogens, and this
Yeah.
actually attaches to attaches to a lot of different DNA bases depending on what species you're looking at. But in human, it attaches to what is called the cytosine DNA base. So there's four. You can think of the DNA alphabet as four letters. So it's
Yeah.
A, G, T, and C. And so the C stands for cytosine, and that's where we usually see the attachment of those methyl groups.
And that is your marker for a change to the DNA based on environment, based on epigenetics.
Exactly. Yeah. I mean, it's very normal to see methylation within the genome. That's in every individual, and we certainly expect to see it at certain areas. But we're really just looking at certain changes in certain areas, and that's also another factor to take into account in these computational models. Is if you have different cells, if you have different individuals that are exposed to different things, this can all make developing a model more complex.
And if you're just, I mean, you're doing all this amazing work, Dr. Wilson, but there's got to be other people like you all over the world working on this with other things in the body besides just placenta. Um,
There are.
oh my goodness, this is so exciting.
Yeah, there are a lot of amazing people. If anyone is interested in placental research, I mean, you can go through a lot of the people I follow, and there's amazing scientists out there and all over the world that are looking at the placenta, that are looking at, I look at the DNA in circulation. Some people look at the cells in circulation, or we have these. I guess, vesicles, you could say, that come from the placenta into circulation, and it carries all the proteins and DNA, and you can get information from those vesicles, and they're called exosomes. Yeah, there's a ton of work being done in terms of looking at changes in the placenta, not only DNA, but changes that we can image on MRI or ultrasound. And there are hundreds of fabulous scientists out there doing that right now. And if you're interested, check them all out because they're very approachable and would love to talk to you about it.
And all of this, the end goal is to decrease complications with pregnancy and increase baby fetal development, like baby health.
Yeah, that's
And of course,
exactly.
mother, right?
Yeah,
Oh,
I
that's
think
awesome.
that's the overarching goal. And I think a lot of people also want to understand, how the placenta develops. I think people, one thing that I think is underestimated is that there's actually a lot of similarities between the placenta and cancer. And I think, we could learn a lot from understanding how the placenta develops and relating that to cancer. And maybe that could even translate into some new cancer treatments or novel understanding of cancer as well, because these things can be all interconnected.
Hmm. It's like a cascade effect. I love it so much. That's amazing.
Yeah, yeah.
Now, I would be remiss before if I didn't ask you about your dog. We heard your dog bark. Your dog's
Yes.
adorable. I love your dog
She
so much.
doesn't like when I give other people attention, so that was her barking at me to give her pets.
Could you talk about your dog?
Yeah, always. Yeah, so I have, she's three now. I can't believe she's three, but this is Frankie. She's named after Rosalind Franklin, who was one of the discoverers of DNA. Yeah, she's a 3-year-old corgi, loves to play, loves belly rubs, not very good at fetch, but that's okay.
Uh-huh. And you have an Instagram account for Corgi, right? Or
I
for
do.
Frankie.
Yes, I
Yeah.
do.
It's Frankie the Corgi or something like that. Did I get it right?
Yeah,
Or
it's
a corgi named Frankie?
a corgi. That's it. Yeah. Yeah.
And he
So
looks
if you
like you
like,
go on all of these adventures, hey?
Oh yeah, we take her hiking a lot and camping when we can. Although this year getting camping sites was a little bit difficult. Yeah, so we try to get out and get her active, but she loves it. Yeah.
Awesome. Okay, so we're at the point where I'm going to open the floor to questions. So if anybody about genetics, I guess what you've been talking about with epigenetics, placental development, I'll open the floor to questions, and people can request to speak. Again, remember, I will double check if you're asking to speak. If I recognize you or you're following me, and you're not like a troll account, because that happens occasionally. And if you are shy, you can, because I don't know, I mean, some people, I'm sometimes I lurk in spaces and not lurk, but I don't want to get speaking privileges, so I'll send the host a message. You can absolutely send me a message, and I'll read it to Dr. Wilson. Okay, so I will give Liz and then Shona ability. So Liz, you're first. Shona, you're on deck, and then there's a question that was sent to me by direct message. Dr. Wilson, are you okay with that?
Yeah, that sounds great.
Yay. Okay. Liz, you're up. Shona, you're on deck.
Yay. Hi, Dr. Wilson. How are you?
Good, how are you?
I'm doing well. So I'm going to ask a question kind of from a psychology standpoint. So obviously with psychology, the argument's always been nature versus nurture, free will versus determinism. And I've been reading a lot recently about epigenetics. And you mentioned like schizophrenia, we know about that in utero and whatnot.
Mm-hmm.
Do you have any kind of thoughts about how we can apply epigenetics to psychology? Is there something?
Yeah, there's a whole field on this, and I actually used to collaborate with a lab in when I was doing my PhD in UBC. So I'm actually going to tell you to check out Dr. Michael Kobor. He's at the University of British Columbia, and he has an entire lab dedicated to that, and a lot of papers on various different aspects of psychology and epigenetics. And yeah, you should really check him out.
What was his name? I'm sorry.
Michael Kobor.
Okay, awesome.
Liz, can I ask you a question? Like your idea is like how the environment shapes DNA and then affects psychology. Is that the idea? Like
Yeah, I'm saying like personality characteristics, zoions, and things like that. But are we guaranteed to just follow our DNA regardless, or is there? I'm trying to think how to put it. I don't know.
nature versus nurture, right?
Yeah, I would say both. I think there's a lot of things that, you read of families with certain history, like health histories. And yes, that might put you at an increased risk, but you can, change what you like exercise, eat well, and reduce your risk. And I think the same goes with DNA, that, nature in a sense, but then yes, you can change these epigenetic marks, and that might influence, your life as well and your health.
Mm-hmm. Cool. Thanks.
No problem.
I have a follow-up question, Dr. Wilson.
Yeah.
So if you can influence through, your environment, these epigenetic markers, does that translate to, what is it called? Like is it germline? Like your ability to pass that on to your offspring? Can you, like, I'm not sure. Have you seen, with Heath Ledger, A Knight's Tale? Have you seen that movie?
Yes, I have. Yeah.
Can you change your stars based on your environment?
Yeah, so there's a idea that's called transgenerational inheritance, and it's the idea, changes that are in you make in terms of your epigenetics and your DNA can be passed down to subsequent generations. And I think there's very compelling evidence in mice. And there is some evidence in humans, but I want to say there's a caveat. So if you're looking at changes in DNA methylation in a pregnant female and you expose them to something and you can see it changes their DNA methylation, let's say, if the fetus is a female, her germline is developing at the same time that she's in utero. So that also means that the grandchild of that pregnant individual is also exposed. And so for me to be fully convinced that transgenerational inheritance occurs in humans, I need to see it in the great grandchildren. And I just haven't seen, for me personally, and this is controversial within the field, any really convincing evidence that that is the case in the human. I don't want to say that it doesn't exist. I just don't know that we know of the mechanism that it would exist today.
Okay, awesome. I believe somebody has a question from direct message that's similar, or it's on this line. So I'll stop asking my own curious questions. Shona, you're up. Thank you for that, Dr. Wilson. And Liz, thanks for the great question. Shona, you just have to unmute yourself. Shona Schneider. Going once, Shona. Okay. All right. Well, on the line of questioning that I had.
Shoot. Okay. I think I unmuted myself.
Yay. Okay. You got it. All right. I'm
sorry.
Go ahead.
Sorry. That was a bad bit of technical terribleness on my part. Okay. I got excited with my question and writing notes, and I forgot the unmute bit. Okay. So you said that the markers that you're looking for are DNA methylation, which normally occurs on cytosine. Is that right?
Yes, that's correct.
Okay, so when you're looking for these predictive markers of changes, how do you know the difference between a normal set of methylation versus something that is unusual or potentially causing these differences that we're talking about?
Yeah, that's a good question. So how we know we're looking at a methyl group versus not depends on the technique that we use to measure it. So some of this has to do with we'll use antibodies that only bind methylation, and we'll pull down all the DNA with methylation. Some of them we do something called bisulfite converting, so it will change all the methylated cytosines to eventually a thymine, and then we can look in that to the reference human genome and tell which was methylated and which was not. And so eventually you're looking at a bunch of numbers on the percent within a sample, the percent of DNA fragments that are methylated at that particular site.
So you're comparing a baseline genome of methylation to your study site, like your study person or sample of methylation, and charting sort of a statistical analysis of the differences between the two.
That's exactly what we're doing, yeah.
Okay, now does methylation occur? Sorry, this was my last question, and then I will give it over to somebody else. Do you see methylation only on cytosine, or does it happen on other base pairs?
It's mostly on cytosine. It does happen on other base pairs occasionally, and on other base pairs in other species, so plants and things like that on different or other model organisms will have the methylation on different DNA bases. But for human, it's 95 or 90% is cytosine.
Okay. I'm not going to ask any more questions. I will write them down and send them in another way and let somebody else have a chance. Thank you so much.
No problem.
I love how excited you are, Shona.
I got very excited very quickly because I teach grade 12 biology and molecular genetics, and I have a page of notes. So
that's hilarious. I was talking to a colleague of mine. I was like, because I'm chemistry and she's the biology teacher, right?
Yeah.
I was like, I'm talking to somebody about epigenetics, and she lost her mind. She's like, that's my favorite part of biology. And then she's like, no, wait. My favorite part of biology to teach grade 12s is actually the reproductive system because they don't know anything.
Yep. And grade 12 is really biochem. It's not really bio, it's biochem. So this has been wonderful. Thank you so much.
No problem. I'm shocked that high school students are getting epigenetics now. That's crazy.
They don't know what's going to hit them. They're going to be so overwhelmed and excited. It's going to be great.
Yeah, I was talking to my colleague, and she as a teacher, like when things change, you model your teaching after the new material. So
you kind of go crazy.
Yeah. Okay, so I'm going to read the question that was sent to me by direct message from Weak Chirp, and it definitely harkens back to the questions I had about can you pass on different DNA based on your environment, and it was, can trauma affect? Is there a link between trauma, like life trauma, or I guess even with mice trauma and epigenetics? Is there anything you found, or is there anything you can tell us about that?
Yeah, I think this is rather controversial. There is an idea that yes, it does. There are changes in DNA methylation that are associated with childhood trauma. A friend of mine, Dr. Nicole Gladish, who's actually in Michael Kobor's lab, just finished her PhD, and so she has a paper out on this where she does talk about some of the changes in DNA methylation that she sees associated with childhood trauma, 20 years after the fact or something.
Okay, awesome. So that was the question from Week Chirp. Paula, you wanted to ask a question or you wanted to speak. I'll go to Paula, and then somebody sent me a message. It's actually one of my students sent me a question. That's awesome. Paula, go ahead.
Hi. All right. Thank you. Hi, Dr. Wilson. Nice to have you tonight. This is all kind of, a new thing for me to listen to. So excuse me if I kind of feel a little naive here. But on your epigenetic pathways, they develop, I would think, in utero. But during your lifetime, can these changes be, like you said, environmentally could change your DNA and stuff? Because autoimmune diseases seem really big in people these days, and I see friends with a lot of conditions like thyroid, or you see a lot of vitiligo, things like that. I don't know if you have any thoughts about that.
Yeah, so two things. Like, yeah, our DNA is kind of solidified in utero, and we do have a lot of like base epigenetic, we call them imprints that are solidified in utero, but uh, epigenetic and those epigenetic marks are very dynamic, so they change over time, and that's not necessarily bad change. They can be good change or just neutral change. But we also have, just because how we, every time a cell divides, replicate our DNA, and so every time we replicate our DNA, there's chance for mutation or variation, let's say, because not all changes in DNA are bad either. And so over time, you can have mutations, variation, changes in epigenetic marks that can all contribute to these areas for sure.
But do you think because our environment can be so volatile that it's increasing in people?
I don't know if I would necessarily make that as a strong, like our environment's changing, so we see these incidences in disease, because I think there's a lot to be said with how we diagnose disease and categorize them has changed. So, we used to put a lot of different people in one category of diagnosis, and now we understand that there's actually different subtypes. And maybe 100 years ago, we weren't necessarily diagnosing these, or hundreds of years ago, we weren't, we didn't have as much cancer because people didn't live as long to, get cancer as it's associated with age. So I think it's a combination of, it could be our environment, it could be the fact that we're living longer, so we see more disease. But I don't think we can say at this point.
Okay, well, thank you very much. Because, like I said, it just seems like these things are increasing. I see it more with people talking about. You kind of wonder what makes your body change. Is it, like you said, because we're living older, or is it something that is environmental that that changed within your body, say in the later years or something? Is that because we're living longer?
Yeah, I think it's a combination of both. Like our environment's constantly changing, and that could be good or bad. We don't necessarily know. And yeah, we're living a lot longer. So I think we see a lot of increase in disease for sure.
Okay, well, thank you very much for answering that. I appreciate it.
No problem.
I was listening to a comedian from Alberta talk about the idea that we just are better at diagnosing these different things. And he made the point that there's probably people with attention deficit disorder, or whatever the more PC name for that is, way back when.
Exactly.
And they just did bad at school, but they were amazing farmers. Like they could just farm all day long, and because they had so much energy.
Exactly. Yeah.
they just weren't diagnosed as they weren't expected to go to school, and they did bad, but man, could they just work the farm. So anyways, I thought that was kind of an interesting point this guy made. Yeah, and I never thought of that we are, if you somehow made it past, like, what hundreds of years ago, what was the age cutoff? Like, if you made it out of childhood, you were pretty lucky.
Yeah, there was like age 5, like there was a
Yeah,
good
age 5.
percentage if you made it 5 that maybe you would make it to adulthood. And
Yeah, that's
yeah,
horrifying.
and like the first
That's
written incidence of cancer, I'm pretty sure was, don't quote me on this, you ask a historian, but I'm pretty sure it was ancient Egypt. It was one of the first written documents that we have of cancer. And so it's not that cancer didn't happen back then, it did, but maybe it wasn't as prevalent because we didn't live as long. And, cancer in younger individuals is still like, it's not as common as you see in older individuals. So when people say, everyone's getting cancer, is it really everyone's getting cancer, or are we all living much longer? And cancer really is just an accumulation of mutations, which, like I said, every time a cell divides, we have a chance of getting a mutation. So if you live long enough, the chances of you getting a mutation that's bad is quite high.
I wish as I get older, I would get mutations to have like laser eyes and things like that. But no, that doesn't seem to happen, does it, doctor?
No, that would be fun. I would go with I would want telepathy. That would be cool.
Telepathy.
Yeah.
Oh, okay. Yeah, some people are like, would they like to know what their animals are thinking? I think that's a dangerous route to go. But anyways,
Just constantly food.
yeah, food. Okay, so I have a question from Karan Deep. Karan Deep is actually one of my students this year,
Oh.
which is awesome. He's in Karan Deep's in the chat today, or in the spaces. He was wanting to know more about machine learning. How does it actually, do your programs actually learn? Do they get smarter as you go, when you're feeding them the data?
Yes, in a sense that you could have models that learn from the data as you go. But the one caveat I want to say is that a model is only as smart as the human that's programming it and the data it's given. So if there's bias in the data that you have, there's going to be bias in the algorithm that comes out. So a lot of algorithms like facial detection, there's a lot of issues with people who do not skin not being picked up. And that's very likely due to the fact that the data that algorithm was given was predominantly on white individuals. And so I think this goes to speak out in terms of representation and having representation of all different humans, just in order to have an accurate machine learning model that is generalizable to all individuals, you need to include all individuals in your original data set.
That's so interesting. I talked about that with oh man, she's amazing, Dr. Shane. Have you heard of Dr. Shane? She's a artificial intelligence programmer.
I feel
Anyways,
like I have. I
she was
recognize the name.
oh, she's hilarious. She wrote a book called "You Look Like a Thing, and I Love You." So she's very tongue in cheek with AI. So she programmed like a little simple neural network to come up with pickup lines by feeding it pickup lines, and it was just spitting out garbage. And the best pickup line it spit out was "You look like a thing, and I love you." So that's the title of her book. And then she was talking about that with me on the podcast, that some hiring companies use these machine learning to sort through all of the people applying for jobs, and that it like totally eliminated all of the women. And they couldn't figure out why it did that, why it was so sexist, because they didn't want to set it up that way. But it just, like, out of the 1,000 applications, any woman just went right into the trash. And they found out that they'd fed it uh, data from their already, like, people they're hired who are all men. And it was like combing through all of the people's resumes for stuff that matched the data. And it was finding, like, this woman played women's soccer or women's field hockey, and it just we don't want that. And this program just eliminated all of these women from contention. And weren't being nefarious or anything, but it was definitely a crazy sexist artificial intelligence program. So that's kind of like what you're talking about. Hey, you got to be careful with the data you
Yeah.
feed it.
Always think about what you're giving it before you make any kind of interpretation on what's coming out.
Right. So I guess with your line of work, if your data set is garbage, man, have you had disasters like that where you've pressed like compute and it's just spit out nonsense?
Oh, for sure. And I mean, and a lot of that's just, you have to spend a lot of time cleaning this type of data. It's not as clear cut as you would hope that it would be. On top of that, like there's a whole issue in as we start generating more and more genetic data, majority of it, I would say, is white. And so that, of course, is not going to be general to the entire world. And so some things I've done in my data is really go seek out some of my more diverse samples, and it's not easy. And so I think as we move forward and as we generate more data, we really got to think about these issues, because what's the point of generating models that are only going to be usable by a certain portion of the population?
That's awesome. Boy, it's just profound. I was another guest on the podcast. He was an informatics, bioinformatics. Uh,
Yeah,
right. And that's what he was talking about is like when you have people come into studies, they're generally more educated, more affluent and white. And that gives you this crazy data set that's just bananas when you try and apply it to the population at large. Huh.
exactly. Yeah.
Huh. Interesting. All right. Okay. Does anybody else have any questions for Dr. Wilson? You just have to ask to be speaker. If you are shy or don't feel comfortable speaking, you can send me a message. Kendra sent me a message about generational trauma. I think we kind of answered that it's you feel it's controversial and the jury's still out a bit.
Yeah, I think the jury's still out on that one. There are studies that show there might be changes, but we don't actually know what those changes mean, or if they are deleterious or coincidental. We really need replicated studies to understand that.
Perfect. Okay, I'll just check my messages. Okay. Give people a second. One more question for you while I'm waiting. Is this area of study with placenta? This is something you feel like decades of work that to be done on it. Like this is like you mentioned. This is like an area you never want to leave.
Yeah, no, it's a very understudied organ, and someone showed a plot once that really stuck with me, in that the only other organ that is less studied than the placenta is the appendix. And which is funny because so much there's a whole field called the developmental origins of health and disease, where a lot of our future health can be related back to the placenta. And so I think for something that is so important, and that you need a functioning placenta in order to have a healthy baby, I think it's a vastly understudied organ. And so I think we need more people studying it.
What what's the most studied organ then?
Oh, I don't know. That's a good
Do you think
question.
like the heart or something like that? I don't
I
know.
feel like the heart would be pretty well studied. Yeah, I don't know. The most studied. I want to say brain is pretty well studied, but it's so complex that there's just so much data out there on the brain as well.
There's a question that just came in. Does your work with genetics, epigenetics, and CRISPR ever come close to touching?
Yeah, so I do have colleagues and friends that do work with CRISPR. And for those that don't know, it's a gene editing tool with the idea of trying to cure some genetic diseases. And so there's a bunch of different people working on this in different fields. And I haven't personally worked with it, but I think in the future I would be interested in working with it, with the idea that if I identify these changes in or DNA methylation, and I want to understand why they're predictive. One of the reasons they could be predictive is maybe they cause the complication. And so in that case, we usually go to looking at cells or looking at animals, and then we might want to do some gene editing to understand what happens when we change the epigenetic signature at specific regions in the genome.
Right. So you see there's a marker, right? There's that methylation, and then you can apply that to an animal group or like even like cells to
Yeah,
see
it just,
what happens, right? And it's just
yeah,
super fast way to do it. Oh, okay. Interesting. Wow, that CRISPR's just amazing, hey?
it is.
I have to like sit down every second and like try to wrap my head around how it works, because
Yeah.
it's super complicated for the average person.
It's still complicated for me.
It's like this found from like some wasn't it some kind of virus that affected bacteria in the ocean? That's how they figured this out, or something like that.
Yeah, it was some sort of bacteria.
Cool. All right. If there does anybody else have any other questions? If not, that's okay. We don't want to string along Dr. Wilson any longer than we need to. Is there anything else you'd like to mention about your work or where people can follow you?
Yeah, so you can follow me on Twitter, and my DMs are always open if you're interested and have any questions to ask me. Yeah, and I always, if you are interested in the placenta or epigenetics, you can go through some of the people I follow, and they're lovely people and all very approachable. So feel free to, send them a DM and chat to them about their work if you're interested in it.
Awesome. One question just came in. I think we'll make it the last one for you. Is that okay, Dr. Wilson?
Yeah, that sounds great.
A question was from Shona. Have you found a specific marker that you feel there's a correlation to a specific condition? Like, are you at that point where you're starting to predictions that you're confident about?
Not yet. And I don't think it's going to be one. I think it's going to be several hundred all in combination.
All
So
right.
it's going to be, I think it's going to be very complex because these are complex conditions. There are conditions where there is single gene, and there is a lot of work on those. But at this point, with how much research has gone on in preeclampsia or preterm birth, I think we're looking at several hundred genes or epigenetic marks.
Okay, awesome. All right, well, thanks for joining us for Spaces Unleashed today, Dr. Wilson. The last bit of this Spaces time is open chat about Bunsen and Beaker. You're welcome to stay for that. If you would like to go right ahead. I mean, I'm so appreciative of you giving up your time to talk. Some folks just they like to know a little bit what's going on with their dogs and with the week. So
That's fair. I'll say goodbye to you, but give Bunsen and Beaker a pat for me.
you betcha. One of them's was around me a second ago. So
Aw.
all right.
All
And
right.
same thing with Frankie. Give Frankie a pet.
I will. Have a good night, everyone.
Okay. Thank you so much, Dr. Wilson. Bye-bye. Oh, that was so interesting, man. I could listen to Dr. Wilson talk about genetics for hours. Okay, so it's overtime or it's Bunsen and Beaker time. If you have questions you'd like to know, or questions you'd like to ask about the week or things that have happened with Bunsen and Beaker, this is the time to ask. And if not, that's okay. We'll end the space a little bit earlier. I had somebody say that I had two different people say that they couldn't hear. Can you guys give me a 100 if you can hear me? Okay, so we've got one. Can you give me a 100 if you can hear me? Okay, we got a couple 100s. Oh, maybe there's lots of people. Oh, okay, they're coming in now. All right, so it looks like it could be an audio issue on those people's end. David, you had a question, so I'm going to give you the ability to speak. And then Bird Like Sofa Tiger, you follow me. You're up next. So David, go ahead.
Oh yeah, that's twice. Oh, I was granted so fast. I almost thought it was somebody else. But anyway, no, I was just curious as to how the fur kids got along at what were they at? Were they at boarding or something like that?
Yeah, it was a kennel or boarding, you betcha. Yeah. And they did. So the place we take to them to, they take pictures of the dogs, which I really like, because we get to see them. Because man, we missed them so much when we were gone. And they have amazing people there that they just love dogs. They play with the dogs. Beaker, I get with the one worker, like the entire day, just was wanting to sit in the person's lap and get pets, fell asleep and just wanted to get cuddled the entire day. So I just love that she had somebody to do that with, because she's a cuddle bug sometimes. And then they have like a big, I don't know if you saw the picture, but they have a big tub for them to get wet in, and Beaker, I guess, was in there like lots. So that's super cute.
Yeah. I saw a bunch of pictures, but those last two, I don't know if you took them or they took them, but they were like, they should go on the calendar.
Oh, yeah, some of them are pretty nice. Hey,
yeah. There's just one of Bunsen, one of Beaker, and especially Beaker. Like, I mean, man, that's just, I don't know if there was just good film or good camera or good whatever, everything. It was quite stunning.
I didn't see the dog pooping in the background of one of the pictures, and I definitely got reminded that there was a dog having a poo in the background of one of them. I think it was the one where Beaker was in the tub. So sorry about that, everybody.
I didn't notice. Thanks.
Oh, you didn't notice? No. Me neither. Me neither. All right. Thanks for the question, David. Does anybody else have any questions about the dogs this week? I believe Kay Lucchini was asking about that, but I don't see Kay Lucchini anymore. There was something came in. What criteria do you use to vet a doggy hotel? Okay, from Too Many Plants. Yeah. So, we were taking Bunsen and Beaker to one kennel. I won't name any of these, and we felt there was just too many dogs. And this other kennel had much less dogs to handlers to space, or more space ratio. And it was recommended. This one kennel was recommended to us by Shar, who is the dog trainer we had on a couple weeks ago, and I literally respect and I trust anything that Shar says. So Shar was the one that recommended this place, and it was absolutely totally worth it. And then the family that runs it, Chris taught their kids as well, so there's a connection to Chris. And I think this place is just amazing. So yeah, so that's to answer that question. That's how we vet it. We by recommendations, and it was recommended by Shar, and Shar is somebody that we really trust because she knows her stuff. Okay. Were there any other questions?
I just had a quick question. So I know they were super excited to see you when you got home, but did they go all cat on you and then realize they were pissed off that you left them and ignored you?
No, they were happy. So my father-in-law, Gord, Chris's stepdad, he went and got them because the kennel was going to close before we got back from the Comic-Con. And so they were in our house, right? So instead of having to go get them, I opened up the door, and we took two vehicles. So I got home a little bit before Chris, and they went bananas. And they were just so happy to see everybody. No, they didn't give us the cold shoulder at all. They're just super happy to see everybody. Beaker literally tried to sleep on top of my face all night. Normally she sleeps kind of at the foot of the bed, but she was like wanting to sleep on top of us. So I think she really missed us, which was pretty cute. All right. Any other questions? Again, you can send one by message if you are kind of shy. And if there aren't any, that's okay. We can end the spaces and call her an evening. All right, going once. Going twice. Next week on Spaces Unleashed, we are having Dr. Sarah McNulty, who is a squid expert. And she also runs Skype a Scientist, which is an amazing organization. So Dr. Sarah McNulty will be on the Spaces next week, and we'll talk every squid. Everything you could know about squid, this amazing person knows. And Sarah's the Squidmobile. So her car, her van, I'm not sure what it is, minivan. It's the Squidmobile, and it's got like a number on the side of it that you can text, and it will text you back a squid fact. It's just amazing. And then the week after that, we've got guests, and we've got some really cool guests, and I can announce that now. We've got next week, we have Dr. Jessica Kania, who is a vet. So if you have questions about your pets, you can ask those in a couple weeks. I was able to book in Jen from The Golden Ratio. So she's going to be chatting with us. I apologize. I keep forgetting her last name. Anyways, somebody will probably send me what her last name is. I apologize for that. And Matt, We Rate Dogs. He's going to be on Spaces Unleashed in the future as well. So he was gracious enough to be a guest, I think later in November. Dr. Katie Mack, also the amazing astrophysicist, agreed to be a guest. So we've got some amazing people coming in Spaces Unleashed all the way into November. So we've grown a really awesome little group here. It's one of the bigger spaces on Twitter, and Twitter's noticing that, which is amazing. So thank you for coming every week. We've been too busy this week to organize the new merch. I know I said that. So this weekend, we will have in the store the 2022 calendar and some of the new adorable merch that we had. We were selling it at the Comic-Con, and it was amazing. A whole bunch of Bunsen and Beaker fans actually found us at the Comic-Con who are local to Calgary. So that was pretty special. We'll chat next week, Tuesday evening. Okay, bye-bye.
Thank you. Bye-bye.
See ya. Bye.